Next, we will be hearing from two of our MITO2i graduate scholarship awardees on their work in diabetes and mitochondria. And first, we're going to have Kassandra Zachos, a PhD candidate by Dr. Jenks' lab and my own lab, and she will be presenting the implications of dietary interventions on mitochondrial dysfunction in type 2 diabetes. Kassandra, over to you.
Thank you, Anna. Good morning, everybody. So today I'm going to talk to you about my project, which is the implications of dietary intervention on mitochondrial dysfunction in type 2 diabetes. So Dr. Harper just gave us a wonderful overview on the biochemistry behind metabolic syndrome, so I'll keep my background brief. But type 2 diabetes is a complex chronic illness with disrupted glucose homeostasis. It is a multifactorial disease, so there's lots of interactions between the genetic and environmental factors, but is often characterized by insulin resistance, which ultimately leads to chronic hyperglycemia. As Dr. Harper mentioned, there are a lot of patient-specific biochemical alterations, so type 2 diabetes looks different on everybody. Type 2 diabetes accounts for over 90% of the diabetes cases in adults, and the number of children with type 2 diabetes has increased dramatically over the last 10 years. Individuals suffering from type 2 diabetes are more likely to suffer from other comorbidities, including anxiety and mood disorders. Beyond the physiological implications, our healthcare system spends over $75 million daily treating diabetes just here in Canada. So how does mitochondrial dysfunction play a role in this? Well, similar to type 2 diabetes, mitochondrial dysfunction can be defined as multifactorial. There are environmental stressors, diet, and the genetics that all play a role in actual function of the mitochondria. And dysfunctional mitochondria can look very different. It can be due to damaged mitochondria, impaired oxidative phosphorylation, a reduced number of mitochondria, or impaired mitochondrial biogenesis. In type 2 diabetes and dysfunctional mitochondria is a bit of a chicken and an egg situation. Is dysfunctional mitochondria actually leading to the onset and the progression of type 2 diabetes, or is type 2 diabetes triggering dysfunctional mitochondria? Ultimately, interrupting this negative feedback cycle is incredibly challenging. But today I'll be talking a little bit about diet and how dietary interventions may be able to do just that. To understand mitochondrial dysfunction in this context, we'll be looking at two specific biomarkers, lactate and circulating cell-free mitochondrial DNA, or CCF mtDNA. On the left side of your screen is an overview of the metabolic pathways that lactate is involved in. Ultimately, when we have dysfunctional mitochondria, we see reduced oxidative phosphorylation or reduced aerobic cellular respiration. To compensate and produce enough ATP, we then have a shift towards glycolysis to help generate enough energy for our body. And this leads to an accumulation of lactate production. Ultimately, that helps feed into the cycle of insulin resistance, increased ROS production, and immunosuppression. On the other hand, CCF mtDNA results from damaged mitochondria. And when these mitochondria are damaged, mitochondrial DNA fragments and gets released into the bloodstream. This then acts as a DAMP, or a damage-associated molecular pattern, and feeds into the inflammation that we see in type 2 diabetes. This mechanism usually works through two different pathways. The first being it'll signal through toll-like receptor 9, which increases NF-kappa B and ultimately leads to increased and pro-inflammatory gene expression. And then on the other hand, it leads to an increase in the formation of the NLRP3 complex, which is tightly linked to mitochondrial dysfunction in many other contexts. However, CCF has yet to be evaluated with dietary interventions in type 2 diabetes. So to summarize, our objective is to gain a further understanding of mitochondrial dysfunction in diabetes and its relationship with aging and dietary intervention, as aging has also been tightly linked to mitochondrial dysfunction. To do so, we will evaluate these two biomarkers in patients that have undergone 2 dietary interventions. On the one arm, we're going to be looking at if CCF levels actually correlate to 3 age-specific cohorts, and we hypothesize that elderly subjects are expected to have higher CCF and lactate levels compared to the other cohorts. And then the other arm will be seeing if the dietary interventions correlate to as high or low levels of biomarkers. And we hypothesize that a low glycemic index diet is expected to have lower levels of these biomarkers. So to summarize our patient population a little bit, you can see more details in the citation below, but overall, to over 2,000 patients responded to recruitment, 658 were screened, and of those screened, 210 were randomized in the study. About 75 and 80 completed the trial following 2 different dietary interventions. I'll explain a little bit more about the diets shortly, but we were able to collect blood samples before the dietary intervention was started at day 0, halfway through the trial, and then at the end of the 6-month dietary intervention. Now, the 2 diets administered, the first being a high cereal fiber diet. So patients were instructed to select the brown option of carbohydrates, meaning brown rice, brown breads, cereals, and so on, and instructed to choose tropical fruit, bananas, pineapples, and mangoes. Now, the low glycemic index diet was patients were instructed to select carbohydrates that don't contribute to a spike in blood sugar. And this included grains like pumpernickels, rye, quinoa, oatmeal, nuts and seeds, and temperate fruits such as apples and pears and peaches. Both groups were instructed to have at least 5 servings of vegetables a day, and were instructed to avoid refined carbohydrates. To do so, as I mentioned, we'll be collecting samples, and that has already been conducted. And once we've done that, the serum and plasma will have been isolated, and we'll be able to measure our biomarkers. Lactate will be measured through a tool called bioprofile analysis, in which it's an actual machine that can give us all of these nutrients and metabolites you see in the chart on your screen. And then for the CCF mtDNA, we will extract the DNA and conduct quantitative PCR analysis to measure the DNA content. Moving forward, we were able to acquire samples from a validation cohort that will then further support these biomarkers and mitochondrial dysfunction in different disease states. These samples have come from the Ontario Health Study, which is an ongoing research study that investigates risk factors and that cause chronic disease. They are one arm of the CanPath study, which is ultimately the exact same thing, but it's cohorts across the entire nation. The diseases we'll be investigating in our validation cohort include type 2 diabetes, cardiovascular disease, and Alzheimer's disease. 100 samples will be collected from each disease, totaling 300 blood samples, and we will only have access to baseline plasma samples. We selected these 2 other diseases beyond type 2 diabetes as they have been already shown in the literature to be associated with these biomarkers and mitochondrial dysfunction. What we like about this cohort is that they also have access to lifestyle behavior, including nutrition data. While they didn't undergo dietary interventions, they do have food frequency questionnaires, and we can assess these individuals' diet and how statistically comparable they are to the interventions that we underwent previously. With that being said, we're at the final stages of the MTA, and then we'll be blindly doing our analyses in a couple weeks and moving forward throughout the rest of the year. Lastly, I just want to discuss the interdisciplinary actions and the importance of our patient engagement. Mitochondrial biomarkers have been applied in many different contexts, especially psychiatric conditions. However, it has yet to be applied in the context of nutritional sciences. As I mentioned, type 2 diabetes is a multifactorial disease, and it requires an interdisciplinary approach to truly understand what's going on here. Beyond this, we are able to disseminate knowledge beyond type 2 diabetes in terms of dietary interventions in other disease states. And from a patient engagement perspective, there is sometimes discrepancy between model organisms, so it's important to work directly with individuals who have the disease that you're investigating. As I mentioned, complex disorders likely require a more personalized approach, and what's more personalized than someone's diet? Diet is often used now as a therapeutic tool in conjunction with pharmacological therapeutics for diabetes, but we could potentially use it as a proactive medicine. And ultimately, we want to ensure that our work is meaningful to the patients. So with that being said, I'd like to thank my supervisor and co-supervisor, Dr. Jenkins and Dr. Andriaza, our lab members, and of course, MITO2i for this opportunity and for the financial support.